Okay, so good afternoon, everybody. Thank you for coming, for your persistence for the last session, which will be exciting as we'll present recent news results from ongoing clinical studies. I'm happy to present as first speaker Dr. Werner Cautreels, who is the CEO of Selecta Biosciences in Boston, and he will report on an ongoing phase 2 trial of lead candidate SEL 212 in development of chronic severe gout. Let's see what this is.
Thank you so much, and thank you, CLINAM, to have me again here this year. So today I'll give you an update on what we are doing at Selecta with antigen-specific tolerance, and I put antigen-specific between brackets because if it's tolerance, then it has to be antigen-specific, right, Jose? So let's talk about this here, the elephant in the room. It's not Beat, but it's not called Beat in this case. It's called actually immunogenicity of biologic therapies. So more and more innovation, more and more new medicines are now biologics. Unfortunately, a number of these biologics are recognized by a percentage of patients as being foreign. And as a result, you can compromise, you make anti-drug antibodies, you can compromise efficacy, you can have a safety risk, and you have unpredictable responses. This is very well known. There are a number of recent publications on this, and we actually have an ally with somebody at FDA, Amy Rosenberg, who is very much involved in this. And we talked to her quite a lot. So what is this about? It's all about the dendritic cell, and most of you know that this dendritic cell is on the crossroad of immune stimulation and immune tolerance. Stimulation, we are very happy about that when we get vaccinations, but we're not so happy about that if there is a drug that the patient is needed and that will start making antibodies against that. So therefore, what we did is we looked into nanotechnology, and we invented this SVP rapamycin particle, I will come back to that when it is co-administered with an antigen, and so through the same route of administration at the same time, then the dendritic cell will go into a tolerogenic state. It will make through the left pathway a population of regulatory T cells, and through that pathway will actually prevent immunogenicity. That's actually the theory. And you will see that both preclinically and clinically we were able to show that. So these particles I will show you a structure later, are co-injected with the antigen. They have to be co-injected with the antigen. You can mix them, or you don't have to put, or not mix them sequentially. And what you see is these particles will just target the spleen and the liver, because that's what particles do. This is not what you like when you do drug delivery, but this is what you like when you want to signal to the immune system. And if you do some histology, and that is maybe difficult to see in this room, is you see that these red-labeled particles actually predominantly go to the dendritic cells and the macrophages, and that is where you would like these particles to be. Now, we have designed these particles to stay intact until they're taken up by the cells. So when you inject them IV or sub-Q, they don't do anything. They only deliver their payload when they're taken up by the cell, and that is actually the key of this. We have done a lot of preclinical work. This has been published, and these are two examples. And what I would like to point out here is that on the top you see that we actually can put the antigen and the immunomodulator inside that particle, and that works. And sometimes you need to do that when you want to protect the antigen from the environment, or sometimes the patient from the antigen, like for instance if this is peanut extract, you don't want to inject free peanut extract into patients. But the problem with that is that if this is a drug, and if that is an existing drug, you have to start all over again. If this is Humira and you put that in a particle, you have to do the whole Humira development all over again. And so what we have shown is that you don't have to put that inside as long as you give it together. But it has to be through the same route administration at the same time, because the same dendritic cells need to see the same signals. And the beauty of this is that you can replicate this technology. We can now, and we have shown that, we can co-administer this with enzymes, with viral vectors, with immunotoxins, and that works. And the beauty of this is that the drug is just a drug you take off the shelf. If that's a new drug, at least you develop the drug, you don't have to develop as a particle. Of course, the rapamycin particle we have developed. The beauty of that is that all the preclinical work, all the manufacturing work, we can replicate over and over again because we have shown that this same particle works with all these different antigens. And the reason for that, you could think about that at least by intuition, is the particle triggers the dendritic cell independently of the antigen. So there shouldn't be a big difference from one antigen to the other. The frequency and how that works is different. As an example, preclinically, and this is published, this is in factor VIII deficient mice. And what you see there is two groups of animals. In black is empty particle with factor VIII and the active particle with factor VIII. And if you co-administer them, in this case we do this 5 weekly injections, a bit like a vaccination schedule. You see that in green that those animals do not develop antibodies against factor VIII with memory. So even though they stop receiving a drug, the particle at week 5, I believe it is, for 6 months they maintain that memory. And that's tolerance, right? Whereas the other animals you see, they develop antibodies. And if you expose those animals to another antigen, they will normally develop immunogenicity. Another example is in gene therapy. And this is where really the elephant of immunogenicity is in the room. What we have shown is that we can prevent further boosting of pre-existing antibodies. We cannot turn around the immune system. We don't kill B cells or T cells, so if you have an immunogenicity, we don't turn that around with this technology. There are ways of doing that, but it's not the basic. We have also shown that we can prevent liver inflammation, and we can also show prevention of formation of new antibodies. This is just an example. This is done with Federico Mingozzi, who is currently actually CSO of Spark Therapeutics, who is a gene therapy company. 2 injections at day 0 and day 21. Day 0 is with luciferase, that's a dummy, and factor IX is what we really want to measure. And what you see at day 21 on the right panel here is that if you measure after the second injection, you do see nice factor IX expression, which you don't have if you had the empty particle, and that is because you have made anti-AAV8 antibodies, enough so that you cannot redose. This is really important in gene therapy because today there is a one-shot chance. If you underdose, the patient cannot be redosed anymore. And if the activity wanes off, you cannot do it anymore. And we have shown that we can do this in an adoptive transfer model, that if we inject in a donor mice AAV8 with or without a particle, and then we transfer splenocytes after cleaning, and we expose the recipient mice to the same antigen, only the animals that have received the antigen and the active particle have received that memory. So we had to go into the clinic. We are not a gout company, but this was a real important product for us, also because it has a high medical need. Gout is a relatively easy disease. If you have too much uric acid, and if you go above 6.8 mg per deciliter, you have crystals, and those crystals go sit in your joints. There's several million patients, 8 million patients in the US. Most of are treated with orals, but if they are resistant to orals, and there are 150,000 of that, they cannot be treated. So the goal is here, you have this magical number of 6 milligram per deciliter in serum, which is also the FDA-approved endpoint, so we don't have to measure pain or anything like that. And the goal is that blue line to bring it down so that these crystals can be eliminated. So here in this case, the drug is the particle with an enzyme called pegsiticase. Why are uricases very immunogenic? Because we do not make endogenous uricase. So pigs don't get gout because they have uricase. If we don't behave like pigs, we may get gout because we don't have the internal endogenous cleanup factory. There is a product on the market called Krystexxa, pegloticase. 90% of patients develop ADAs, and that results in like infusion reactions, anaphylactic reactions, and everything else. So we did a phase 1b study in about 60 patients. We have patients that receive just the enzyme in red, only the particle in blue, was first in man, or then the combination in green, and we did dose ranging. And it's a very simple picture. If you only give the particle, nothing happens, and all these patients, they're going to stay above 6. If you give the enzyme, it works, so the curve goes down, but after 2 weeks, you see it go up again. And it's only if you give this mitigation of immunogenicity that you can prevent that, and you can have a monthly drug. And if you compare then the 3 critical parameters, on the left, you have ADAs. In red, you see them go up. No ADAs on the bottom. You don't have the enzyme on the top anymore. You have it still on the bottom in green. And therefore, serum uric acid levels are fully controlled. So we have done that. But that's after a single administration. What you want in these patients is a 6-month treatment. And so we now are in the process of doing this phase 2 trial. We're actually completing this in about 150 patients, which means that we have 150 patients that received like 5 infusions of this. In this slide, I show you where they received a combination in the orange part, and then we challenge them with only the enzyme at the end to see if we have that memory. And indeed, we do have memory, but not really enough. We knew that this worked in non-human primates. In humans, also, I'm kind of a candidate for this drug. My thymus is not extremely active anymore, so age actually is very important as a factor. So we are now doing 5 and 6 co-administrations, and then we'll see for the memory. The data I show here was presented at the EULAR in Amsterdam earlier this year. And what we showed is that if you go into the active dose range, that about 80% of patients remain controlled. That if you would just give the enzyme by itself, you see only 1 patient in 6 or 1 patient in 7 would still meet that, because everybody makes ADAs, and that results from the 2nd injection and the 3rd injection in a loss of activity. So what is next? We are completing this phase 2 trial. I can maybe later on comment a bit on safety. This is actually very safe. We don't see a lot of infusion reactions or anaphylaxis. And if it happens, it's the first time. We do pre-medication. And one of the biggest parameters, which is, I think, important for you to know, maybe the biggest parameter for safety is the speed of injection. And that is probably not a surprise. The number of particles that you get over a unit of time in the beginning is really important. So we will go into phase 3. We have announced that we will do this the end of the year. It means that, I don't know if the FDAers are still here. We have a lot of interactions with FDA, very good, in terms of whether that's preclinical talks, GMP. I think we fixed everything. We make proposals, we go discuss with them, they make good counter proposals, and then whenever we execute what we agree on. And if you do that, you're fine. We are not a gout company. We have a number of products in, one more in the clinic, which is LMB100. Actually, on the bottom you see, LMB100 is a mesothelin targeted immunotoxin that the company next door here, Roche, had licensed from NCI, had tried to optimize B-cell and T-cell epitopes, and that doesn't work. And so they gave it back to NCI, you know, we licensed that from NCI, we combine it with our particle, and we are in the clinic, and hopefully next time, May 2020, we can present on that. And gene therapy is a really important area for us, and we do have a license agreement with Spark. So I think we always thank our patients and all clinical trial sites, investigators, and in this case, because we believe they make the big elephant a bit smaller. So thank you for that.
So thank you very much for this really interesting presentation. I'm sure there are some questions.
Very interesting presentation. So rapamycin mTOR inhibitor, so are there, what about the other analogs, or what about other ways to intervene in that particular pathway in terms of the outcome that you describe here?
Yeah. We, and that is first our founder in vitro, Uli von Andrian at Harvard, and then we in vivo, we tried all kind of immunomodulators, and we have actually IP on that. Rapamycin came systematically out as the very best. by a long shot. And of course, yes, the mTOR pathway and dendritic cells, I'm not an expert in that, but there is a logic in that. And so we have focused on this. But there are other ways of doing it, but rapamycin so far is the best. If you try to do this yourself, it took us about 3 years and 650 different formulations and combinations for this to happen, because it sounds trivial on paper, but it's not really in practice. Jose.
Okay, there's one more question.
So thank you very much. It's really fascinating, and I'm very happy that we are getting close to proof immune tolerance in humans. So you allude to the dosing. So can you give some advices in how to test? Because I always believe, as you mentioned, that the frequency of the treatment is as important or more than not the concentration of the reagents that we are using in this particular approach.
Yes. So what we have shown preclinically is that the same particle at the same doses works. And we believe that is because the dendritic cell needs to receive enough for it to go into that stage. The dose we give therapeutically is going to be about 10 milligrams a month, which is about kind of the loading dose of rapamycin. But we don't give anything every day. We just do it once. We believe the frequency of how many vaccinations, how many vaccinations you need is going to be dependent on the antigen. And that depends on the indication. So in gene therapy, our plan is to give it each time, because you give gene therapy today and then maybe in 6 months and then again. If you would give Humira or something chronic like enzyme replacement therapy, you may need more frequent vaccinations to maintain that memory. And then you may need to actually give a booster or a negative booster every, 6 months or a year to maintain that memory and to keep that population of Tregs big enough.
Thank you again for this very interesting presentation. We come now to the next speaker, who is Christiane Rieken.